We'll move on to enzymes, and enzymes are mainly proteins, and they are reaction specific. That means that one enzyme will act on only one substrate, and an example is that I have here is carbonic anhydrase. This is present in red blood cells, and what it does is it speeds up the reaction between water and carbon dioxide, and this produces carbonic acid, and it can go in either direction. And so because it's present in red blood cells, this reaction occurs really fast in the red blood cells. The same reaction occurs in plasma as well, but because carbonic anhydrase is not present there, the reaction does not occur as fast. So you can see one of the main properties of enzymes is to speed up reactions. However, they do not themselves get changed during the reaction. So as seen in this picture here, you can see what the enzyme does is it combines with the substrate, that means the substance it's going to act upon, and they form a complex, and somehow there's an internal rearrangement, and after the rearrangement the enzyme is liberated, it's free in order to take part in the reaction again, and you get the end product over here. So that's how these enzymes act. Some enzymes need activation, not all of them. One example is Pepsinogen, which is present in the stomach. It is an enzyme which helps with protein digestion. Now, Pepsinogen gets activated to pepsin in the presence of hydrochloric acid, and when if hydrochloric acid is not present, then Pepsinogen will not get activated. And this is the reason why the stomach is acidic in nature. The medium inside stomach produces hydrochloric acid so that this pepsinogen can be activated. Let's move on to nucleic acids, which are extremely large molecules, and what they contain are nucleotides, which are made up of nitrogen bases, pentose sugar, and a phosphate group. And the nitrogen bases are adenine, which is the A here, thymine, guanine, and cytosine. And we have another one not shown here, which is uracil. So let's look at the two different types of nucleic acids. DNA is deoxyribonucleic acid, which is present in the nucleus, and this is a double-stranded. As you can see, it has this double helical property. These are the four nitrogen bases which are present. It carries out protein synthesis, so it gives orders for protein synthesis. It contains the hereditary material in it, and so it's like the bandmaster which gives orders, and so it gives orders for protein synthesis present in the nucleus. RNA is ribonucleic acid. So RNA is ribonucleic acid, which is present outside the nucleus. And in this, the nitrogen base thymine is replaced by uracil. And there are different types of ribonucleic acid. You have mRNA, tRNA, and so on, but we won't go into those details. But this is single-stranded, so doesn't have this double helical structure, and it takes orders for protein synthesis. So those are the differences from DNA. So takes orders for protein synthesis. Let's then move on to ATP, which is the short form for adenosine triphosphate. It is an energy molecule, and the only utilizable form of energy which the cell can use. So while we take in glucose or lipids or proteins, you cannot use glucose directly. Glucose or lipids or proteins have to be converted to ATP, and then that ATP is used to provide energy. So ATP, because it has 3 phosphate molecules in the bonds there, that's where energy is stored. So when ATP loses one of the phosphate bond, it releases energy which the cell utilizes, and so it becomes ADP, which is adenosine diphosphate. And then if it loses another bond and more energy, it becomes adenosine monophosphate. So the body the cells utilize the stored ATP, and then when you run out of ATP stores, then more is made up from the fuel that you eat, which is glucose, lipids, and proteins. So here, let us look at this video which explains this really well.
The molecule used by most organisms to transfer energy is called adenosine triphosphate, or ATP. An ATP has 3 parts: adenine, ribose, and phosphate groups. Adenine is one of the bases found in DNA and RNA. Ribose is a sugar molecule found in RNA. The adenine and ribose portion of the ATP molecule is called adenosine. The 3 phosphates linked to the ribose in a chain are called triphosphate. Adenosine, together with the 3 phosphate groups, is called adenosine triphosphate, or ATP. Notice the wavy lines between the 2 outer phosphates. The wavy lines represent high-energy bonds. Breaking the first high-energy bond in ATP releases energy and a free phosphate. This energy is used to power cellular activities. Adenosine, with only 2 attached phosphate groups, is called adenosine diphosphate, or ADP. Energy from the breakdown of food is used to change the ADP to an ATP. The energy stored in ATP is transferred and released when needed to power cellular activities. This process may occur as many as 10 million times per second in some cells.
So you can see how when we eat something, when we eat food, that goes to produce ATP, which is then utilized for all our cellular activities in the body. And then once ATP is, the amount is diminished, then you go and again you eat more, and then again you make more ATP. So you can see it's a constant cycle that occurs in our body.